So we will learn about protein translation. You know how the message encoded in the DNA or in our gene get translated into the protein for the function in our body. So before starting, please look at here. You can see I have drawn a structure of DNA from where the mRNA is derived in the process of transcription, and that mRNA encodes all the message of a gene that will be translated into the protein. So overall, from the transcription to the translation process, it is called as central dogma of molecular biology. So although this is a quite schematic theme I have drawn here, the translation of protein from the gene via mRNA is a quite tricky and complex process. It involves a lot of biochemical and enzymatic reactions. So basically, we will discuss what are the components those involved in the protein translation. We will not describe about the transcription event. We will focus on the protein translation event. So what are the most important molecules or enzymes required to conduct the protein translation event? So I mention here foremost important things to conduct the protein translation event are the mRNA, that is the gene, ribosomes, (70S and 80S) ribosome. 70S stand for the ribosome system of prokaryotic system, and 80S is for the eukaryotic system. The tRNA, there are few other things including the initiation factors, elongation factors, and release factors. So although the role of lot of enzymatic molecules and biochemical molecules occurred, the most emphasized component of the protein translation machinery are I have enlisted here. One is gene, that is mRNA, that encodes the message, the ribosome that holds the mRNA to carry out the process, the tRNA that contain the anticodons and that brings the required amino acid to the mRNA, and the initiation factor that initiate the protein translation activity, and then elongation factors. Once initiation happens, during the protein translation, there is a elongation of the protein chain, and once the protein translation event occurred or concluded or terminated, it need to be released from the ribosome and other components, so it needs the release factors. So let's draw how protein translation event happens. So this is the 5' end of the mRNA and this is the 3' mRNA. The 3' end contains the poly A tail. And towards the 5' end of the mRNA, the ribosome attach to this part and reads the start codon of the mRNA. You know, the mRNA contains all the message for the protein in the form of triplet codons, and you must be knowing about this. So first codon will be the ATG, or in the mRNA T will be replaced by U, or you can say AUG. And in few cases, if there will be no AUG start codon, there will be GUG and UUG. Another two codons also used as start codon in the protein translation machinery. So what will happen? In the ribosome, the small subunit will attach with the mRNA, then it will implement the tRNA. The tRNA, as I mentioned, the tRNA contains the anticodon here in the anticodonal loop and a CCA tail. Okay, the CCA tail, and in this region the amino acid will attach. But for most important thing is that which amino acid will be attached to this end. It's depend upon the anticodon. For example, if there is AUG codon in the mRNA, its cognate anticodon will be here in the tRNA, and that will bring the methionine amino acid to get attached here in the start of the protein translation machinery. Subsequently, the protein translation will continue and it will read as a triplet codon, and for each and every next codon, if it will be a different codon from the start codon, here a different amino acid will come, and subsequently the protein chain will continue like this. And during this event, during the initiation, there will be initiation factors. The initiation factors helps to initiate the protein translation event, and once the initiation happens, there will be elongation of the translation system, and during the elongation there are the elongation factor like elongation factor TU, elongation factor S like this, and subsequently this event continues, and once the protein synthesis get complete, the ribosome will be halt and the ribosome will stop the protein synthesis event. And how the molecular system will know that there is stoppage of protein synthesis? Because it's depend upon the presence of the stop codons. There are 3 stop codons. If the translation system finds the presence of stop codons like UAA, UAG or UGA, if any of the codon will be there, there will be stoppage of protein synthesis event, or you can say the protein synthesis event will stop there. And once the protein synthesis event stopped, it need to release the components. The components are ribosome, the elongation factors, the tRNA and all these things. So the release factors, in short it is called RF, they will play their role to release the ribosome system from the translating mRNA. And during the protein synthesis event, as I mentioned, the chain will continue like this. But protein synthesis is not only the addition of consecutive amino acids. Sometimes the synthesizing protein get modified co-translationally and post-translationally. So once the protein synthesis began, the synthesizing protein undergo modification. The modification can be co-translational modification or post-translational modification. Co-translational modification means the modification of protein or synthesizing amino acid will occur simultaneously. Once it attaches to the polypeptide chain, immediately it will undergo modification. And post-translational modification means the protein will continue to synthesize, it will finish its synthesis, and then it will undergo modification and it will conduct its function. So modifications are 2 types. One is co-translational modification and another is post-translational modification. So in the modification process, what get modified? Some synthesized proteins having very short chain, we can say them as polypeptides, and some acquire a particular structure and shape. Until and unless a protein undergo its transformation to make its proper structure, it will not function properly. So structure of the protein is directly correlates with the function of the protein. So once the amino acids made the chain and the polypeptide is formed, to have a particular shape, it needs some chemical modification to maintain or make the structure. And the structure, as I mentioned, it can be co-translational or post-translational. So what are the common modifications we find in the protein? The common modifications are addition of methyl group, that is CH3, addition of OH group or hydroxyl group, addition of sulfur group, addition of carbohydrate or addition of lipid with the protein. So, once the protein got synthesized, basically it will go modification. There are several lot of other modifications, and I mentioned the most important a few basic modifications that we can find in our body system, and they are addition of carbohydrate, lipid, addition of methyl group, addition of OH group, and addition of sulfur group. So these protein modifications lead to the functional activities or helps to develop the protein quite correctly to bring its function very well. Usually the methyl modification leads to develop the protein in such a way that there will be quite less enzymatic degradation or breakdown of proteins, so that it will withstand the stress condition in the body, so that the loss of protein does not happen and body will maintain a particular function quite effectively. And these protein synthesis events usually occurs in the cytosol of the cell, and more importantly when it comes to the ribosome events, the endoplasmic reticulum encoded with lot of ribosomes. So endoplasmic reticulum is also a very effective organelle that is associated with the protein translational event, as it encodes a lot of ribosomes. And as far as protein translation is considered, as I mentioned about the prokaryotic and eukaryotic protein translation event. The protein translation in the eukaryotic system is quite rapid than the eukaryotic system. So in the eukaryotic system, so there will be 6 to 8 amino acid addition will occur during the protein translation event per second. But in the prokaryotic system, it will be greater than 16 to 17 amino acid per second. So in the prokaryotic system, the protein translation is quite effective and it gives a high frequency addition of the amino acid to the translating polypeptide compared to the eukaryotic system.